Stand in front of a mirror and draw an imaginary line straight down the middle of your body. At first, the design looks almost suspiciously tidy. 2 eyes, 2 ears, 2 arms, 2 arms, 2 legs, 1 nostril on each side, which feels like a strange thing to brag about, but anatomically, congratulations. Now move closer. 1 eyebrow probably sits a little higher. 1 side of your smile pulls differently. 1 ear may stick out more. Flip a photograph of your face, and suddenly the person you have seen every day looks like a relative who knows all your passwords. Then look beneath the skin, and symmetry completely gives up. Your heart sits mostly left of center, with its tip usually pointing left. Most of your liver occupies the right. Your stomach and spleen are on the left. Your right lung normally has 3 lobes. Your left has 2. Your intestines do not hang like a neat bundle of identical tubes. They rotate, coil, and anchor in a directional pattern. So here is the puzzle. If symmetry is so useful that evolution gave you matching limbs, paired eyes, and a face organized around a midline, why did it build your insides like someone packed a suitcase during an earthquake? The obvious answer is that bodies are messy. Things grow where they fit. One side gets used more. Small mistakes accumulate. That answer is not completely wrong. It is just explaining the least interesting part. Some of your asymmetry is random. Some develops through habit, injury, or ordinary variation. But the deepest asymmetries in your body are deliberate. Long before you had a face, fingers, or a functioning heart, your embryo had to perform an extraordinary trick. It had to decide which side would become left. And the strangest part is that choosing the familiar direction may be less important than making sure every organ follows the same decision. To understand that, we need to separate 3 things that people casually throw into one bucket. First, there is programmed asymmetry. This includes the normal positions and shapes of your internal organs. These differences are built into development. Second, there is functional lateralization. Your 2 sides may look similar while doing different jobs. Hand preference and some patterns of brain activity belong here. Third, there is ordinary individual asymmetry. The tiny differences produced as billions of cells divide, move, respond to forces, and grow in a world that never treats both sides perfectly equally. A left-pointing heart, a right-handed tennis serve, and a slightly crooked smile are all asymmetries. But they are not the same biological problem. Start with the first one, because it begins when you are less like a tiny person and more like a construction site, with no walls. An embryo needs coordinates. Cells must know where the head will be, where the tail end will be, which surface faces the back, and which faces the belly. Without those axes, development would be less build a human and more release several organs and hope they network. Left and right are especially peculiar because, unlike head and tail, the 2 sides initially look almost equivalent. A cell cannot consult a compass. It cannot look around and say, the liver goes over there. I remember it from the diagram. Somewhere, symmetry has to be broken. Much of what we know about that first break comes from animal models, especially mice, combined with human genetics and clinical evidence. That distinction matters. Scientists cannot casually watch every molecular event inside an early human embryo. But when experiments in mammals reveal a mechanism, and human mutations affecting the same machinery produce predictable laterality disorders, the pieces become much stronger together. In mammals, a temporary structure near the embryonic midline acts as a left-right organizer. In the mouse, this organizer includes a region called the node. Its cells carry microscopic projections called cilia. You have probably heard of cilia in the respiratory tract, tiny moving structures that help carry mucus and trapped material out of the airways. Embryonic nodal cilia are not literally the same cilia relocated from your future nose, but they use related cellular machinery, and some of them perform a completely different job. They rotate. Because the rotating cilia are tilted, their motion generates a directional flow of fluid across the organizer, usually toward the embryo's left. This is not a dramatic current. There is no microscopic whitewater rafting team inside you. It is a tiny physical bias in a tiny space at exactly the right developmental moment. But a bias is all the embryo needs. there is another beautiful detail hidden in that sentence. Leftward flow is only meaningful because the embryo has already established other directions. The cilia are positioned and tilted with reference to the head-tail and back-belly axes. Their rotation is therefore converted into a consistent sideways current. Left is not discovered in isolation. It is calculated from coordinates the embryo has already built. That makes the process less like choosing between two identical doors and more like completing a three-dimensional map. Once you know forward, backward, top, and bottom, rotation can produce a reproducible sideways bias. Development builds orientation on top of orientation. A mistake in an earlier coordinate system can therefore echo into later anatomy, which is one reason congenital disorders of body patterning rarely behave like a single misplaced sticker. Experiments in mice helped reveal how important that flow is. When researchers disrupted proteins needed to build or move nodal cilia, the usual left-right arrangement could become randomized. Even more strikingly, experimentally reversing the direction of flow could reverse laterality signals. That gave developmental biology something it desperately wanted: a bridge from physics to anatomy. Rotation creates flow. Flow creates a left-sided difference. That difference activates a molecular program. The program guides organs into asymmetric forms. Except one piece of the bridge is still under construction. Researchers continue to investigate exactly how cells read the flow. One possibility is that the current transports signaling material preferentially toward one side. Another is that non-moving, sensory cilia detect the mechanical force and trigger changes inside the cell. Recent work has strengthened mechanical sensing models, but the full story is still being refined. This is important because popular explanations often turn the node into a magical drainpipe. Water goes left, therefore heart goes left. Biology is more irritating than that. The flow is an early cue, not a tiny hand physically shoving organs around. Downstream of that cue, a signaling network becomes active on the left side of the embryo. Important members include nodal, lefty, and a gene regulator called PITX2. Think of them less as workers carrying organs and more as a system that marks territory, reinforces the signal, limits it, and tells developing tissues which version of their instructions to use. PITX2 is sometimes described as a marker or executor of leftness. That is useful shorthand, as long as we do not pretend one gene designs the entire left half of a human. Organ development involves many pathways: local forces, tissue interactions, and organ-specific instructions. The left-right network gives them directional information. It does not personally install the plumbing. And then the quiet molecular difference becomes visible. The early heart begins as a relatively simple tube near the midline. A straight tube cannot become a compact, chambered pump with correctly connected vessels by simply getting larger. It must bend and loop. In normal human development, the tube bends in a characteristic direction during the 4th week, helping position the future chambers and outflow tracts. The gut also refuses to remain straight. It lengthens faster than the space immediately available to it, temporarily projects into the umbilical region during normal development, rotates, returns, and becomes fixed in a particular arrangement. The liver grows predominantly to the right. The stomach and spleen take up their usual positions on the left. The lungs develop different lobar patterns. Notice what is happening. Your organs are not manufactured as finished objects and then placed into an empty torso. They grow while touching, pulling, folding around, and connecting to other tissues. Position changes shape. Shape changes. Connection. Connection changes. Function. The body is not packing a suitcase. It is sewing the suitcase while everything is already inside. This is also why saying that an organ is merely on the wrong side can be misleading. Laterality signals influence morphogenesis, the actual process by which tissues acquire form, not just location. A heart can point right yet remain internally coordinated. Another heart can remain left of center while its chambers, valves, or great vessels connect abnormally. The address does not tell you whether the building was wired correctly. The same principle applies to the lungs and gut. A left-sided organ identity includes features of branching and shape, intestinal rotation, is a sequence, not a final pose. By the time an anatomy textbook gives every structure a colored label, the real developmental drama is already over. That explains how asymmetry forms. But why have asymmetry at all? For the outside of an actively moving animal, bilateral symmetry is extremely useful. A body with a front end can move through the world in a chosen direction. Paired limbs can generate balanced forces. 2 eyes and 2 ears can compare signals arriving from different positions. A central midline provides a stable reference for muscles, skeleton, and nervous system. Bilateral symmetry was a major evolutionary development in animal body plans. But bilateral never meant every structure had to be duplicated. It means one plane can divide much of the body into broadly corresponding left and right sides. Internally, many organs are singular, elongated, branching, or connected into loops. A digestive tract has to travel from entrance to exit. A heart must connect incoming and outgoing vessels. A liver can perform its work as one large organ. There is no general engineering rule saying that all of these structures should be duplicated just to satisfy a mirror. Asymmetry can also help differently shaped organs coexist in limited 3-dimensional space. But we should be careful here. It is tempting to invent a perfect adaptive reason for every detail. The heart points left because left was obviously superior. The liver sits right because evolution ran a spreadsheet, and the spleen was assigned the remaining desk. In reality, evolution works with inherited developmental systems. Once a coordinated arrangement functions, later changes occur within that existing architecture. Some asymmetries may improve packing or performance. Others may reflect ancient developmental constraints. For many individual left-right details, scientists can explain the mechanism more confidently than the ultimate evolutionary reason. That distinction how versus why is where a lot of neat stories quietly become fan fiction. Now we reach the part that overturns the whole puzzle. Most people have the standard organ arrangement, called Situs solitus. But some people are born with Situs inversus totalis, in which the major internal organs are arranged as a near-complete mirror image. The heart points right. The liver is predominantly left. The stomach and spleen are right. The lung pattern is reversed. And many of those people live without physiological problems caused by the reversal itself. Some discover it only during imaging, surgery, or an examination, when a clinician goes looking for a familiar landmark and finds that the map has been flipped. So apparently the universe does not collapse when the heart points right. This tells us something profound. The normal direction is highly conserved, but for many aspects of organ function, coordinated arrangement matters more than the absolute direction. A fully mirrored system can still connect coherently. Imagine mirroring the entire road network of a city. It would look bizarre to anyone who knew the original map. But traffic could still flow. Now imagine mirroring 3 intersections, leaving the bridges unchanged, rotating the airport, and moving the hospital into the river. That is closer to the problem in heterotaxy. Heterotaxy is not simply the organs are backward. It describes abnormal, discordant arrangements in which structures may show mixed, ambiguous, or duplicated, right or left-sided features. The liver may lie near the midline. The spleen may be absent or divided into several smaller spleens. Both lungs may resemble the usual right lung, or both may resemble the usual left. Major veins and arteries may connect abnormally. Complex congenital heart defects are common. This is why complete reversal can be less disruptive than incomplete coordination. One alternative blueprint can work. Several blueprints used simultaneously are much harder to connect. There are exceptions and complications, of course. Situs inversus can occur as part of a broader condition, and people with it still need clinicians to know their anatomy. But mirror anatomy by itself is often compatible with normal life. Heterotaxy, by contrast, often creates serious medical problems because the issue is not cosmetic location. It is mismatched identity and plumbing. The contrast gives us a natural experiment. It suggests that the embryo does not merely stamp left onto one half and walk away. It must communicate that choice across multiple organs, each with its own developmental program. So the heart, vessels, lungs, gut, liver, and spleen build a mutually compatible system. And this brings us back to cilia. Primary ciliary dyskinesia, or PCD, is a group of inherited disorders involving the structure or function of motile cilia. In the airways, those cilia normally help move mucus. When they do not work properly, people can develop chronic wet cough, persistent nasal and sinus symptoms, recurrent ear disease, and progressive lung problems. Related structures are also important in reproduction, so fertility can be affected. But some of the same molecular machinery is used by the embryonic cilia involved in left-right patterning. According to the current Gene Reviews summary, complete mirror image organ reversal occurs in roughly 40 to 50% of people with PCD. Heterotaxy occurs in about 9 to 12%. Those numbers are not random trivia. They connect a cellular motor, an embryonic flow, organ laterality, and adult respiratory disease in one evidence chain. They also reveal a crucial nuance. If nodal cilia fail to provide the normal directional bias, the embryo does not necessarily become symmetrical. Often, laterality becomes less reliably directed. Other developmental processes can still produce asymmetry, but the usual orientation is no longer guaranteed. It is like removing the arrow from a compass, not erasing the landscape. The historical term Gardner syndrome was used for a combination involving chronic sinus disease, bronchiectasis, and Situs inversus. Today, PCD is understood as genetically diverse, with many causative genes and a range of presentations. Not every person with PCD has reversed organs. Not every person with reversed organs has PCD. And noticing that your face looks uneven in a bathroom mirror is not a reason to diagnose yourself with a ciliopathy before breakfast. Your body contains more than one system for making left and right different. The brain proves it, structurally and functionally. The two cerebral hemispheres are not identical. Language is left lateralized in most people. Some aspects of attention and spatial processing often show rightward biases. Motor control crosses sides, so the left hemisphere largely controls the right side of the body, and vice versa. This real science was flattened into one of neuroscience's most successful bad personality quizzes. Left brain people are logical, right brain people are creative. Why lateralized brain functions at all? One proposal is that specialization can reduce duplication and allow the hemispheres to process different kinds of information in parallel. Comparative research shows that behavioral and neural lateralization is not uniquely human. It appears across many vertebrates and even some invertebrates. But here again, mechanism is firmer than the grand evolutionary story. Lateralization can be measured. Its exact advantages can depend on the task, the species, and the environment. So the honest conclusion is not that the left hemisphere evolved for language in one clean step. Human language likely recruited and reshaped older, already lateralized neural systems. Evolution renovates. It rarely clears the lot and starts with a fresh architectural brief. That is not how a functioning brain works. Complex tasks recruit networks across both hemispheres. Lateralization is a matter of relative specialization, not two tiny employees fighting over whether you should become an accountant or buy watercolor paint. Handedness is also more complicated than it looks. A genome-wide study involving more than 1.7 million people identified dozens of genetic loci associated with left-handedness or ambidexterity. That is strong evidence against a single left-handed gene. Hand preference is polygenic and shaped by development, with each associated variant contributing only a small part. Handedness and language lateralization are related, but not locked together. Most right-handed people have language functions biased toward the left hemisphere. Yet most left-handed people also show left hemisphere language dominance. Left-handers are more likely than right-handers to have atypical or more bilateral language organization. But left hand does not simply equal right brain language. Now add the strangest clue. People with complete visceral reversal do not automatically have mirrored minds. Brain imaging studies of people with Situs inversus totalis have found that some structural features, such as the broad twist, known as brain torque, may reverse more often. But language lateralization and handedness do not reliably flip with the organs. In one imaging study, language-related lateralization in people with complete visceral reversal looked broadly similar to matched controls. The sample sizes in this research are necessarily limited because Situs inversus is rare. Still, the pattern is clear enough to reject the cartoon version. Visceral laterality, brain structure language dominance, and handedness overlap in complicated ways, but they are not controlled by one universal left-right switch. Your body does not choose left once. It negotiates left and right repeatedly at different scales, using partly different mechanisms, which finally brings us back to your face. A mild degree of facial asymmetry is normal. Studies using three-dimensional imaging find measurable left-right differences, even in people selected for ordinary, apparently symmetrical faces. These differences can be only a few millimeters, invisible until someone draws lines across a scan or flips a photograph and ruins your afternoon. Some asymmetry comes from developmental noise, tiny variations as cells divide, migrate, and respond to local signals. Genes influence the growth system, but genes do not specify the exact final position of every molecule. Biology produces robust patterns without manufacturing two digitally cloned halves. Other differences can emerge or change during growth. Teeth, jaws, muscles, soft tissues, posture, injury, and patterns of use may all contribute in particular cases. But be suspicious of anyone who looks at one photograph and confidently announces the single habit that caused a person's face. Reviews of facial asymmetry repeatedly note that many proposed behavioral explanations are difficult to prove without good longitudinal evidence. A body is a history, but it does not come with captions. This also means symmetry is not a simple health score. Severe or newly developing asymmetry can matter clinically, especially when it affects function. But ordinary mild asymmetry is not evidence that someone is genetically inferior, poorly developed, or losing a contest against their pillow. Perfect external symmetry would be biologically unusual. Perfect internal symmetry would require a radically different animal. Your body is better understood as a layered compromise. At the largest scale, bilateral symmetry gives you a balanced frame for moving through the world. Beneath that frame, a conserved developmental program assigns left and right identities, so organs can bend, rotate, and connect coherently. Within the nervous system, different functions develop population-level lateral biases without obeying one master switch, and across your face and limbs, small variations record the unavoidable flexibility of growth. Programmed asymmetry. Functional lateralization. Individual variation, 3 phenomena, 1 body. So stand in front of the mirror again. The face looking back at you is not a failed attempt at perfect reflection. Behind it is a heart that once began as a tube and folded in a particular direction. A gut that rotated. Lungs that developed different shapes. A brain whose 2 halves divided some of their labor without dividing you into 2 personalities. And all of it traces back to a stage when your embryo was still establishing its coordinates. Before you had a name, it had a midline. Before you knew left from right, it had to discover the difference. And perhaps that is the real answer to why human bodies are asymmetrical. Symmetry gives a body balance, but asymmetry gives it direction, specialization, and room to become a working whole. The goal was never to build a perfect mirror. The goal was to build a body that knew which way to turn.